Hello everyone, we are studying the volumetric analysis in the paper first of S.Y.B.Sc. So in this chapter we have studied the types of the volumetric analysis, which are: first is the neutralization titration, second is the complexometric titration, redox titration and precipitation titration. Now in the last lecture we have studied the types of the neutralization titration. We know that the neutralization titration, it is nothing but the titration of the acid and bases. So in that case we have studied basically three types of the curves for the neutralization titration. First is titration of strong acid versus strong base, second is strong base versus weak acid, third is weak base versus strong acid, and fourth one is the weak acid versus weak base. So we know that the different three types of the three curve are obtained from the first three types of the neutralization titration, and fourth one, it is practically not applicable because weak electrolytes, we know that they cannot dissociate completely in the aqueous solution. So this is the first type of the titration or the titrametric analysis. Now for this lecture we are going to study the next type, second, it is the complexometric titration, and then further the redox titration. So let us start with the complexometric titration. So name indicates that complex formation takes place in this type of the reaction. We know that in titration we are adding some first electrolyte in the burette or the strong electrolyte in the burette, and the substance whose concentration is to be determined, it is taken in the conical flask. So in this titration we are using first substance is the metal ion and second is the ligand or the complexating agent is used. So first solution is the metal ion solution and second one something complexating agent. The solution of the complexating agent is used so that the metal reaction between the metal ion and the complexometric reagent or chelating reagent, it is also called as the chelating agent. So complexating agent or the chelating reagent reacts with the metal ion and it forms the stable complex. So depending upon the stability constant of this complex, the corresponding precipitation may take place or generally change in the physical change in the solution takes place, or color change depending upon the indicator, it will take place at the end point of the titration. So in this case we are going to study the reaction of the metal ion with the complexating agent. Now in this titration, generally the complexating agent, it is taken in the burette. So solution of the complexating agent or chelating agent, it may be taken in the burette, and then in the conical flask metanyl solution is taken whose concentration we can determine, or the corresponding complex formation. After complex formation at the particular concentration of the metal ion only, at the particular concentration of the metal ion and the chelating agent, the complex formation or stable complex is formed at the end point of the titration, and it can be indicated or it can be detected by the presence of indicator which shows the change in the color. Now there may be change in the color in the form of solution, or solid may be formed, or colored solid may be formed. So depending upon the indicator. Now in this type of the titration, we know that the stable complex is formed. So first there is, if you consider this type of reaction, this is the metal ion, M stands for the metal ion. Now this metal ion complex or the solution is there and it is bonded to the n number of the water molecule. So this is simple, the general example of the complex. In this complex metal ion is present and it is bonded to some ligands. We know that in the complex there are different geometries are given for the complex. Square planar geometry is there, then tetrahedral is there, octahedral is there. So generally when 6 ligands are present, the geometry will be the octahedral geometry. So in this case, the complex at the center, metal ion is present and it is surrounded by the ligands. So these ligands are nothing but the donor atoms or the group. They will form the coordinate bond with the metal ion. So these ligands or these ligands are the donor group. These are nothing but the donor group. It may be the atom or it may be the group. We know that the donor group, it may be electron rich species that may having the negative charge, or it may contain the lone pair of the electron. So for example, we can consider oxygen, sulfur, phosphorus, nitrogen, halogens. All these contains the extra pair of the electron. So this can be easily available for the donation to the metal ion and it will be form the stable complex. Secondly, negatively charged ion may be present that may act as the ligand. So ligands are nothing but the species that are electron rich species, and they will donate the electrons to the central metal ion and they will form the stable complex of the particular geometry. Now when we consider this reaction, this metal ion contains this H2O. Now this H2O are nothing but the ligands which are present in the complex. So what happens if we add the another ligand like L? So depending upon the affinity of the L, if it having the strong affinity for the metal ion, then it will replaces these ligands which are present in the complex and this ligand will get removed. So this water molecule will get replaced by the ligand here. So if we consider n number of the water molecules are present in this complex, one ligand is added here. So this ligand will replaces the one of the H2O molecule. So we are getting MH2O n minus 1L. So what happened here? In this complex, one H2O molecule is get replaced by the another ligand. So this type of reaction, this is the H2O molecule which is removed from the complex and one ligand is get added in this complex. So in this way what happens, this procedure or this process may get continuous, replacing all the H2O molecule by the ligands. So what we are getting, MLN. So all the n number, whatever the valency of the metal ion, depending upon that, the n number of the ligands will get replaced here. H2O molecule will get replaced by the stable ligand. So we are getting the stable complex. So depending upon this affinity of this ligand towards the metal ion and the stability of the complex, we are getting the stable complex at the particular concentration of the metal ion and the ligand. So formation constant are very important in such type of the reaction. So what is our target? We can determine the metal ion concentration by forming this stable complex in this titrametric analysis. So this type of titration, it is called as the complexometric titration. So complexometric titrations are nothing but the reaction of the or titration of the complexating agent, solution of complexating agent with the metal ion solution to form the stable complex at the end point. So this type of titration are called as the complexometric titration. Now in this titration, different types of the indicators can be used. We know that in the titration, indicators having very important role, they will shows the physical change or color change at the end point. And depending upon the type of reaction, different indicators can be used. Now, the indicators used here are Paton is one of the indicator, readers, these are the indicators that can be used. Further, in this case, the different types of the indicators like KMnO4 and potassium dichromate, these two are the strong oxidizing agent, we know that. So KMnO4 or potassium dichromate, this type of indicator may be used or it may be used for this in the type of the titration where they acting as the oxidizing agent. So in this case, So in the next point we are going to study the redox titration. We know that the redox titration contains this transfer of the electron. So in case of this type of reaction, either redox titration K2Cr2O7 and KMnO4, this type of the indicator are used. But in this case we are using pattern and reader, this type of the indicator. Along with this we can also use the another indicator, it is ureochrome black tea, and another is the solochrome black tea. Solochrome black tea. So instead of these two, pattern and readers, these are not much used in the titration, but the ureochrome black tea and solochrome black tea, these two indicators are generally used in the complexometric titrations. So these are rare, generally they are not used, so we are using these two indicators for the titration. In this case now we can plot here the graph for this type of titration. So in this case the graph of PM versus 0.01 molar EDTA solution. So this is the graph of PM. So metal ion minus logarithm of the metal ion concentration is nothing but the PM. PM equals to minus log of metal ion concentration. So negative log of metal ion concentration plotted against the EDTA. Now this EDTA is the chelating agent, very important chelating agent. We will after this graph we will see the structure of this EDTA. It is the ethylene diamine tetraacetic acid, EDTA. So this is the structure of the EDTA we have shown here. Ethylene diamine tetraacetic acid. So generally it is available as the sodium salt or the stable sodium salt. We can make it solution and we can use it in this type of titration. Now if we see the structure, this is ethylene molecule, it is bonded to 2 nitrogen, therefore ethylene diamine, and tetraacetic acid means 4 acetic acid molecules are attached to nitrogen, here 2 and here 2. Now ligands are categorized depending upon their donor atoms. How many donor atoms are present in the ligands? Depending upon that donor atoms, it can be categorized as monodentate ligand, didentate ligand, bidentate ligand, tridentate ligand, or the polydentate ligand. So in this case, how many donor atoms are present here? We know that in complex, ligands are acting as the donor atoms. It will donate the electrons to central metal ion and form the coordinate bond. Here nitrogen containing lone pair, so 2 donor atoms here, and again these 2 carboxyl groups in the acidic medium it will dissociate to form COO minus. So this carboxylate ion is formed and it also acts as the donor atom for this type of reaction. So how many donor atoms are present here? 4, carboxylate ion 4 and these 2. So 6 donor atoms or the 6 donor groups are present in this EDTA. So it is act as the hexadentate ligand. It is called as the hexadentate ligand. So this EDTA, it will form the stable complex or stable octahedral complex with the different types of the metal ions like copper, zinc, etc. So or chromium. So this type of the metal ion will get complexed with this type of the reagent. It is called EDTA. So it is also called as the chelating reagent or the complexating reagent. Now let us see the graph. So in this graph, what we have taken? We have plotted the graph PM, that is minus logarithm of the metal ion concentration with the 0.01 molar of the EDTA solution. So how this EDTA solution is prepared? It may be prepared by direct method or indirect method. So in the direct method, the particular weight of the EDTA, it is taken according to molarity and it is diluted in the water. Or in the indirect method, first approximate solution is prepared and it is titrated with the another substance to get the exact normality. So in this case, the 0.01 molar EDTA solution is taken and the metal ion solution is taken. So this EDTA solution is taken in the burette and metal ion, for example copper metal is there or the zinc metal is there. So this metal can easily form the stable complex with the EDTA. So these metal ion solutions are taken in the conical flask. Now when we study this type of titration, from the burette, EDTA solution is slowly added in the metal ion solution. At the end point, there is change in the color is observed. So depending upon the indicator. So for this type of titration, we can use the ureochrome black T indicator. It is generally used, and this indicator will shows the change in color. So it will shows the wine red. It will forms the wine red color solution at the end point. So at the end point, stable complex is formed. So when this stable complex is formed, then only we are getting the change in color. Now if we study the change in the metal ion concentration here with the EDTA solution. So at first this graph, it will starts from the 2 value of 2, it is the metal ion concentration. It will starts from the 2 value and it is straight here. There is no increase in the concentration of the metal ion at the start. When we are adding the solution up to nearly 10 ml or 9.6 or something, 9.6 to 7 ml, up to this there is no much change in the metal ion concentration is observed, and there is sudden change in the concentration is observed nearly 16 to 18, and we are getting this value 10 raised to 16. So this 10 raised to 16 at this point 10 raised to 10, this will indicate the corresponding concentration 10 raised to 6, 10 raised to 4. So this graph will indicate it is very much different from the neutralization titration curve. So in this case, sudden change is observed. There is no curve or there is no continuous change. Sudden change is observed in the concentration. So depending upon the formation constant of the complex, we are getting this type of value. Now let us see the structure of the EDTA, how it forms the complex. Now this is the structure of the octahedral complex which is formed of the EDTA, which will form the EDTA with the central metal ion. So this having the octahedral geometry. So we can see this structure. This nitrogen, it is bonded to the metal ion at these two sides of the square planar geometry at these two corners. Then the two sides of this octahedral complex, these are occupied by the carboxylate ion, oxygen of the carboxylate ion, and the remaining upper side and lower side, it is occupied by the another two carboxylate ion. So there are 6 donating atoms are present in this type of this geometry, that is the donor group, which are present in the EDTA. So this stable octahedral complex formation indicates the end point for this titration, and depending upon that value we can determine the metal ion concentration for this type of the titration. Now in this case, this titration, it is applicable to determination of different types of the metal ion concentration. For example, in case of determination of the hardness of the water, we know that the simple water contains the very less amount of the dissolved ions, inorganic compounds, or the dissolved ion like calcium, magnesium, chloride ions. The different types of the ions are present, but when concentration of the calcium ion and magnesium ion, it will get increased. So what happens in that case? The water, it becomes hard. So if it becomes hard, we cannot use it for the drinking purpose. So in order to determine the exact concentration of the calcium ion and magnesium ion, we can determine the total hardness of the water by using this type of titration, that is the complexometric titration. So in that case, water sample is taken and in the burette EDTA solution is filled. Zinc sulfate may be used for this type of titration, and EDTA solution, it is titrated with the hard water sample, and according to the end point or the calcium and the magnesium it will form this stable complex with EDTA at the end point, and depending upon that value we can determine the concentration of the calcium and magnesium which are present in the hard water. So this is the one of the application of the complexometric titration. So in the next lecture we will study the redox titration and the precipitation titration. Thank you.